Hello everyone, my name is Ziwan Poon from University of Malaya. On behalf of our project members, today I would like to share some key highlights on our work on the phylogeography and population genetics of epiphytic filamentous red algae on eucheumatoids. The eucheumatoid seaweeds often host a variety of epiphytic filamentous algae or EFAs, which are generally thin and small-sized red algae that grow on and to some extent into their tailline. So the EFA abundance fluctuates throughout the year and they seasonally develop into blooms, infecting the eucheumatoids. And this causes problems for the farmers since the excessive growth of these epiphytes will reduce biomass production and the quality of carrageenan obtained from the raw material. It is likely but not certain that the transfer of eucheumatoids among countries has led to the introduction of EFAs in regions where they are non-native, and this will be investigated through this study. Despite being a relevant problem for eucheumatoid farming, the species level identity of EFAs has barely been studied. The first stage of this project is to obtain a better understanding of the diversity and distribution of the EFAs at local and global scales using morphological and genetic tools. And this actually contributes to ongoing efforts of GSSTAR projects. Then the second phase of this study is to identify the origin and dispersal pathways of these EFAs. So we'll be selecting 1 to 3 EFAs growing on farm seaweeds and the surrounding habitats, and we analyze their population genetic structure. This map shows the sampling sites of where we collected samples of EFAs from farm and wild or natural populations of eucheumatoids. So the samples were collected from regions where farm eucheumatoids are native, and that includes Malaysia, the Philippines, and Indonesia. And we also collected samples of EFAs from where farm eucheumatoids are introduced. So these include Tanzania, Madagascar, and Fiji. In Malaysia, we collected EFA samples from 8 farm sites and 6 natural populations, as shown in this map, while in the Philippines, samples of EFAs were collected from 2 farm sites. As for Indonesia, samples were collected at 5 farm sites and 1 natural population. Moving on to the Western Indian Ocean and Pacific Islands where farm eucheumatoids are introduced, we collected samples from 3 farm sites and 2 seagrass beds in Tanzania, 4 farm sites and 2 wild populations in Madagascar, and 1 farm site in Fiji. In this study, we used DNA barcoding with the RBCL and COX-1 markers to identify the EFAs growing on eucheumatoids from the native region and regions where they were introduced for farming. So DNA sequencing confirmed that the common epiphytes in Malaysia include species of Serranium, Galeella, Chondria, Tolypiocladia, Lavelia, and many more. At present, 64 RBCL barcodes and 52 COX-1 barcodes have been generated, and lab work for a few more samples are in progress. Within the Malaysian samples, we found that Melanotamus thailandicus, which is previously known as Neosiphonia thailandica, is the most common and abundant species of EFA. And this is confirmed through morphological analysis and DNA sequencing, as you can see here in this RBCL phylogenetic tree. The Melanotamus samples are resolved into 3 clades, and the first one corresponds to the sequence of the holotype, first reported to grow epiphytically on Gracillera fischeri in Thailand. So it appears to be a host generalist. Another interesting finding is that although Melanotamus specimens in clades 1 and 2 are found on both cultivated and natural populations of eucheumatoids, those in clade 3 are restricted to the wild eucheumatoids from Malaysia. So based on the genetic differences, this is very likely a new species of Melanotamus, and it remains unknown whether the same species is present on wild eucheumatoids in other regions. From this collection, a small subset will be selected for genome skimming based on the yield and quality of genomic DNA isolated. This is just to show the genetic structure indicated by the RBCL and COX-1 genetic markers. So effectively, we found higher diversity and density of the EFAs on the farm versus the natural populations of eucheumatoids for all three species of epiphytes. And we also observed a rather similar diversity of EFAs between the farmed and the wild eucheumatoids, but as I mentioned earlier, Melanotamus plate tree was only found on the wild host. A subset of the samples was sent to CFAS, where one of our partners conducted histological work on the samples to look at how the epiphytes penetrate into the host tissue. The common epiphyte in the Philippines is identified as Melanotamus thailandicus based on morphological characterization and some initial sequencing results. So sequencing for the rest of the samples is in progress. And Joseph has conducted histological studies on most of the samples collected. So moving on to samples from Tanzania, Madagascar, and Fiji. In SAMS, Dr. Bracco and Callum have actually isolated and dissected over 1,000 epiphytes from farmed and wild populations of eucheumatoids and also those found on seagrasses from surrounding farm. So from these collections, they detected 6 morphotypes of epiphytes, and they include Melanotamus, Herpesiphonia, Serranium, Cetroceras, Galiella, and cyanobacteria based on morphological characterization. Several samples from Paje, Tanzania, were confirmed as Melanotamus thailandicus clade 2 from 5 RBCL sequences and 2 COX-1 sequences generated. So at the moment, multiplex barcoding with the RBCL marker is currently in progress, and Dr. Bracco is also waiting for the sequencing results from 11 samples that submitted for genome skimming. So we are very much looking forward to those sequencing results. During the course of examining the EFA samples, we also look into their mode of attachment and their abundance. So for the samples from Western Indian Ocean and Fiji, we noted that the majority of samples belong to the Melanotamus morphotype, so it's almost 70% of the samples, and this is followed by members of Ceramiaceae, which is 13.8%. So the Melanotamus are firmly attached to the host, so this means that they brings the most damage to the eucheumatoid host. While for the Ceramiaceae, they are less tightly or firmly attached to the host. This project has enabled a better and current knowledge of the diversity of epiphytes on eucheumatoids in Malaysia, Tanzania, the Philippines, Madagascar, and Fiji. And this effort is not possible without the close collaboration with our local partners. Apart from that, we also identified Melanotamus thailandicus as the dominant, common, and most widespread epiphyte associated with farm eucheumatoids in Malaysia and the Philippines, which are the native region where eucheumatoids are farmed, and also in Tanzania, where eucheumatoids are introduced for cultivation. Results from this study provides us with some insights into the spread of this epiphytic pest, which will be useful in developing biosecurity best practices and policies. So the infestation of the epiphytic pest is a recurring and persistent issue in eucheumatoid farming, and this is expected to worsen with climate change as observed in Tanzania. New data from this study will also be used to populate the open access online Atlas of Algal Diseases, which will be available to a broad audience worldwide, including those from the academic, industry, and regulators. This project enabled the training of 3 early career researchers in taxonomic study of this difficult taxonomic group. In Malaysia, one graduate student and a technical assistant were trained in molecular biology skills and to identify the EFA morphologically. I would like to end my presentation by acknowledging all these parties who have helped with sample collection. Thank you very much.